Can epigenetics treat schizophrenia? Have you ever wondered if there is a way to treat schizophrenia by changing how our genes work without altering the actual DNA? This is where epigenetics comes into play. Epigenetics looks at reversible changes that affect gene expression. It does this through processes like DNA methylation, which adds chemical groups to DNA, and histone acetylation, which modifies the proteins that DNA wraps around. Both of these processes can influence how genes related to brain function are activated or silenced. Schizophrenia is a complex mental health condition influenced by various genes and environmental factors. Research shows that epigenetic changes can play a role in the development and symptoms of schizophrenia. These changes can affect genes involved in neurotransmitter systems, such as dopamine, glutamate, and gamma-aminobutyric acid. These neurotransmitters are crucial for brain signaling and often show irregularities in individuals with schizophrenia. For example, abnormal patterns of DNA methylation have been found in genes that regulate inhibitory neurotransmitters, which may contribute to the symptoms of the disorder. Current medications for schizophrenia, like Clozapine, which is often used for treatment-resistant cases, seem to influence epigenetic regulation. This might help explain why they work for some patients. Some studies suggest that combining Clozapine with medications that modify epigenetic marks, such as Valproic acid, could improve treatment outcomes, especially for those who do not respond well to standard therapies. There are two main types of epigenetic treatments being researched. The first is DNA methyltransferase inhibitors, which aim to reduce DNA methylation. This could potentially reactivate genes that have been silenced in schizophrenia. The second type is histone deacetylase inhibitors, which increase histone acetylation. This can enhance gene expression and may help correct the epigenetic issues linked to the disorder. Since epigenetic modifications are reversible, they present attractive targets for new treatments. However, researchers still need to identify which specific epigenetic changes should be targeted and how to tailor these treatments to individual patients. More studies are necessary to find specific epigenetic markers associated with different symptoms and responses to treatment. This could lead to more personalized therapies in the future. In summary, while epigenetics shows promise for developing new treatments for schizophrenia by correcting gene expression abnormalities, dedicated epigenetic therapies are still in the early stages of research. More work is needed to make these approaches clinically viable.